Welcome to the Deep Dive, where we dig into complex sources to give you, well, the shortcut to really getting it. And today, we're tackling a really big question, maybe the fundamental question for life. How do living things create order, grow, reproduce, when everything seems to tend towards chaos?
It's all about energy, right? And how cells manage it.
It really is a profound question, like you said. Every single living thing, it's basically an energy master, constantly, battling that chaos.
Exactly. So for this deep dive, we're getting into the nitty-gritty of metabolism, mostly drawing from Chapter 13 of Lehninger Principles of Biochemistry, our mission today. Untack how cells handle their energy budget. Look at the chemical reactions making life tick. Understand ATP, that universal energy money. See how electrons power, well, pretty much everything. And finally, marvel at how exquisitely metabolism is controlled. Prepare for some real aha moments about what keeps you going. Okay, let's start with the basics. The cell's energy budget. This field is called bioenergetics. It's essentially the study of how living systems manage energy transformations. Right, how they capture, convert, and use energy to do all the work of staying alive. Growth, movement, reproduction, all of it.
What I find amazing is that all this complex biological stuff, it still follows the basic laws of physics, right? The laws of thermodynamics.
Absolutely, no exceptions. The first law is straightforward.
Yeah.
Energy is conserved. You can't create it or destroy it, just change its form or move it around.
Okay, simple enough. But then the second law says things tend towards disorder, towards entropy.
That's the one. The universe as a whole is always getting messier, more random.
So here's the puzzle. How do cells, which are incredibly organized structures, maintain and even increase their order without breaking that second law? It feels like they should fall apart.
It does seem paradoxical. Yeah. But the key is that cells are open systems. They aren't isolated. Think of it like this. They constantly exchange energy and matter with their surroundings. They pull in energy-rich stuff, like nutrients or sunlight.
Use it to build and maintain order inside.
Exactly. And in the process, they release waste heat and less ordered molecules back out. So the total entropy of the cell plus its surroundings increases, satisfying the second law.
Right.
Critically, they are never at equilibrium. Equilibrium for a cell means death.
Okay, so to really get this, we need some terms straight. Free energy.
That's the energy actually available to do useful work under cellular conditions, constant temperature and pressure. If a reaction releases free energy, its AG is negative. We call it exergonic. It can happen spontaneously.
And if it requires energy input, AG is positive, it's endergonic, needs a push.
Right. Then you have enthalpy H, which is basically the heat content. Reactions releasing heat are exothermic.
And those taking up heat are endothermic, and entropy S is just the measure of disorder or randomness.
And they're all linked by that key equation, AG turns 7S. A negative NAT, a spontaneous reaction, usually happens if heat is released. Negative 8 or disorder increases. Positive use or some combination.
Now, here's a really important point, something that often trips people up. The difference between standard free energy change and actual free energy change.
Yes, this is crucial. AG degrees is a constant. It's measured under very specific standardized conditions. Think pH 7, 25 degrees Celsius, 1 molar concentrations. Useful for comparison, like a benchmark. It tells you the potential of a reaction, and it's directly related to the equilibrium constant, AG degree.
If kg degree is greater than 1, the reaction favors products, AG degrees is negative. If less than 1 favors reactants, AGD is positive.
But the actual AG, that's the one that matters in the dynamic environment of a real cell. It depends on the actual moment-to-moment concentrations of reactants and products and the temperature.
So what does this mean for you listening right now? A reaction might look unfavorable in a textbook positive GD degrees. Maybe it wouldn't naturally go very far on its own. But inside your cells, if the products are constantly being whisked away and used up,
or reactants are being supplied,
then the actual G can become negative. The reaction gets pulled forward. Think Le Chatelier's principle from chemistry.
Exactly. The cell manipulates concentrations to make things happen. Which brings us to another key idea. Additivity of free energy changes.
Meaning you can just add them up?
Pretty much.
Yeah.
For reactions that happen in sequence, the overall free energy change is the sum of the individual steps. And this is how cells perform biochemical magic. They couple an energetically unfavorable reaction, endergonic positive G, with a highly favorable one, exergonic negative G.
Like making glucose 6-phosphate, which needs energy.
Plus 13.8 kilojoules, unfavorable.
To breaking down ATP, which releases a lot of energy.
Negative 30.5 kilojoules, very favorable.
So you link them together and the overall reaction becomes favorable.
Precisely. Glucose plus ATP gives glucose 6-phosphate plus ADP. The overall GMO is now negative 16.7 kilojmol. It goes.
Wow. So the cell is constantly doing this clever chemical accounting, linking reactions together. Okay, how do cells actually do these reactions?
Yeah.
What's in their chemical toolbox? It can't just be random bumping around.
Definitely not random. There's a deep chemical logic. And while there are thousands of reactions, they mostly fall into just 5 general categories. Evolution is efficient.
5 main types. What are they?
Things like making or breaking carbon-carbon bonds, internal rearrangements, isomerizations, free radical reactions, though maybe less common than others, group transfers, very important, and oxidation reductions, redox reactions.
Let's touch on a couple of principles. How do bonds actually break?
2 main ways. Homolytic cleavage is where each atom gets 1 electron, makes radicals, usually unstable. More common in bio is heterolytic cleavage, where 1 atom gets both electrons from the bond.
Leaving one atom electron-rich and the other electron-poor, creating ions like carbanions or carbocations.
Exactly. And those are typically very unstable on their own, which leads to the second principle, the interaction between nucleophiles and electrophiles.
Electron donors and electron acceptors, the basic push-pull of bond formation.
That's the heart of it. Now think about carbon-carbon bonds, the backbone of life. Making or breaking them seems tricky because of those unstable intermediates you mentioned.
So how does the cell manage it?
This is where things get really clever, especially involving carbonyl groups that see double bond O. The oxygen pulls electrons, making the carbon slightly positive and electrophile. But crucially, that carbonyl can stabilize a negative charge, a carbanion on the adjacent carbon.
It sort of spreads out the negative charge, makes it less reactive, like resonance stabilization.
Exactly. It provides a temporary parking spot for those electrons. This trick is used all over metabolism, in things like aldol condensations or decarboxylations. Think of the aldolase enzyme in glycolysis.
So the cell strategically places a carbonyl group to make a difficult reaction, well, possible. Smart. What about those internal rearrangements?
Those involve shuffling electrons around within a molecule, changing its structure without changing the overall oxidation state, like glucose 6-phosphate turning into fructose 6-phosphate in glycolysis. Same atoms, just rearranged.
And group transfers, you said they were especially important.
Absolutely central.
Yeah.
Transferring acyl groups, glycosyl groups, but especially phosphoryl groups of the PO3 aerobit.
Usually from ATP. Right.
Very often. Attaching a phosphoryl group to some intermediate molecule often activates it. It's like giving it a jolt of energy, making it ready for the next reaction step.
So phosphorylation primes the pump, so to speak.
That's a good way to put it. And the enzymes that transfer these phosphate groups of ATP are called kinases. You'll see them everywhere in metabolic pathways.
Okay, let's zoom right in on ATC, adenosine triphosphate, the cell's energy currency. What makes it so special? Why ATP?
It's not really about having high-energy bonds. That's a bit of a misnomer. Breaking any bond requires energy input. The large negative free energy change when ATP is hydrolyzed to ADP and phosphate, pi, or to AMP and pyrophosphate, PPI, comes because the products are much more stable than ATP itself.
More stable?
Yeah. Several reasons. First, you relieve the electrostatic repulsion between the negatively charged phosphate groups crammed together in ATP. Second, the free phosphate ion, pi, has better resonance stabilization than when it's part of ATP. And third, the products, ADP and pi, are better solvated, better surrounded by water molecules.
So it's like releasing a compressed spring. The energy comes from relaxing into a more stable state.
Exactly. And get this, the actual free energy released inside a cell, the phosphorylation potential, PP, is often even more negative than the standard value, negative 30.5 kilojmol.
Why is that?
Because the cellular concentrations of ATP, ADP, and pi are nowhere near the 1M standard condition. ADP is kept high, ADP and pi relatively low. Plus, magnesium ions bind to ATP, affecting its properties. In a real human red blood cell, for instance, the actual GP might be closer to negative 52 kilojmol. That's a significant difference, a much bigger push.
Wow. Are there other molecules that pack a similar punch, other high-energy compounds?
Oh yes, some pack even more. Phosphenol pyruvate, PEP, is a big one. Dersch agrees around metagide 61.9 kilojmol. Part of that comes from the product immediately converting to a more stable form. 1-gamma-3 bisphosphoglycerate, negative 49.3 kilojmol, is another key intermediate in glycolysis. And phosphocreatine, negative 43.0 kilojmol, acts like a quick-access energy reserve in muscle and brain.
For rapidly making ATP when needed.
Precisely. Even thioesters, like acetyl-CoA, negative 31.4 kilojmol, are considered high energy because their sulfur-carbon bond is less stable than the oxygen-carbon bond in a regular ester.
Okay, so when ATP provides energy, it's not just randomly breaking down near a reaction. How does it actually transfer that energy?
That single arrow you often see, ADP plus pi, is almost always shorthand for a two-step
Two
process.
steps.
Yes. First, a part of the ATP molecule, usually a phosphoryl group, PO33, sometimes a pyrophosphoryl, PO3, or even an adenyl group, AMP, is transferred to the substrate molecule or to an enzyme. This transfer activates the recipient, raising its free energy content.
Makes it less stable, more reactive.
Exactly. Then, in the second step, that activated intermediate undergoes the desired reaction, and the transferred group, pi, p pi I, or AMP, leaves.
Ah, so ATP lends its energy by first modifying the reactant.
That's the key mechanism. For instance, activating fatty acids involve transferring AMP from ATP. The pyrophosphate, p-pi, that's released is then immediately broken down into two phosphates, providing an extra energetic pull, making the whole process highly favorable.
This activation strategy must be fundamental for building things, moving things.
Everything. Synthesizing DNA, RNA, proteins, pumping ions across membranes against their concentration gradients requires huge amounts of energy. Muscle contraction changing protein shapes, all driven by ATP group transfers.
And it's not solely ATP doing this, right? Other
No.
similar molecules.
Correct. Other nucleoside triphosphates like GTP, UTP, CTP are energetically equivalent. Their terminal phosphate bonds release similar amounts of energy. And cells have enzymes like nucleoside diphosphate kinase that readily swap phosphates around like ATP plus GDP, ADP plus GDP to keep all the NTP pools balanced.
So the energy captured mainly as ATP can be easily distributed.
Yes. And you also have systems like adenylate kinase, 2-ADP, ADP plus AMP, helping buffer the energy state, and as we mentioned, phosphocreatine in muscle, providing that rapid phosphoryl reserve.
One last fascinating point about ATP. It's thermodynamically unstable, wants to break down, but it's kinetically stable, doesn't just fall apart in water.
Right. It needs an enzyme catalyst to lower the activation energy barrier for hydrolysis or group transfer. This kinetic stability is essential for regulation. Energy is only released when and where an enzyme allows it.
Okay, if ATP is the currency, what's the engine generating it? You mentioned redox reactions, electron transfers.
Indeed. The flow of electrons is the fundamental source of energy for most life. Think of electrons moving from reduced fuel molecules like glucose or fatty acids, ultimately offered to oxygen. This flow releases energy, like water flowing downhill generates power. This electron flow, this difference in electron affinity between molecules, creates an electromotive force that the cell harnesses to do work, primarily making ATP.
How do we keep track of these electron movements?
We usually talk about half reactions. One substance loses electrons, it's oxidized, and it's the reductant or reducing agent. Another substance gains electrons, it's reduced, and it's the oxidant or oxidizing agent.
I remember oil rig from chemistry class.
Oxidation is losing, reduction is gaining, still works. In biology, oxidation often involves losing hydrogen atoms dehydrogenation, since a hydrogen atom is just a proton and an electron. We track the oxidation state of carbon atoms, for instance, by how many bonds they have to more electronegative atoms like oxygen. More bonds to oxygen means more oxidized.
And electrons can move in different ways, not just solo.
Right. They can move directly as electrons, as hydrogen atoms, Hu plus E, as hydride ions, Hu, which is a proton with 2 electrons, or even by direct combination with oxygen. We use the general term reducing equivalent to cover any of these forms of electron transfer.
How do we measure the tendency for electrons to flow?
Using the standard reduction potential, EU degrees, measured in volts. It reflects a substance's affinity for electrons under standard conditions, pH 7. A more positive EU degree means a stronger pull for electrons, a better oxidant. We can use these EUG values to calculate the standard free energy change for a redox reaction using the equation EOU a degree, where N is the number of electrons transferred and F is the Faraday constant.
So a larger difference in reduction potential between the electron donor and acceptor means a more negative AGU degrees, more energy released.
Exactly. The bigger the voltage drop, the more energy is available. And crucially, cells don't release this energy all at once, say, in the burning of glucose. That would be wasteful, like an explosion.
They do it stepwise.
Yes, through metabolic pathways. Electrons are passed along in a controlled manner, often to specialized universal electron carriers, which conserves the energy efficiently.
And the main carriers are?
The big two families are nicotinamide nucleotides and flavin nucleotides.
Let's start with the nicotinamides, NADO and NADPO.
Right. Derived from the vitamin niacin. They are water-soluble coenzymes that move electrons around the cell. They accept 2 electrons and 1 proton, typically as a hydride ion, becoming NADH or NADPH.
And they have different roles.
Generally, yes. NADH is usually involved in catabolism, breaking things down to generate ATP. Think glycolysis and the citric acid cycle feeding electrons into the respiratory chain. NADPH is typically used in anabolism biosynthesis, like making fatty acids or steroids where reducing power is needed.
That's a key distinction. And NAD has other jobs too, doesn't it? Not just redox.
Fascinatingly, yes. It's used as a substrate by enzymes like DNA ligases in some bacteria, and importantly by sirtuins, which are enzymes involved in regulating proteins via deacetylation. Sirtuins impact inflammation, aging, gene expression, big areas of research now.
Wow. So NAD levels affect more than just energy metabolism, and the niacin deficiency causing pellagra dermatitis, diarrhea, dementia really drives home how vital these carriers are.
Absolutely. Then you have the flavononucleotides, FMN and FAD, derived from riboflavin, vitamin B2.
How are they different from NAD and ADP?
A key difference is that they're usually very tightly bound to enzymes, sometimes covalently, acting as prosthetic groups. We call these enzymes flavoproteins, another big difference. Flavins can accept either one electron or two, forming stable intermediate radical forms, semikinos. This makes them very versatile, able to mediate electron transfers between donors that give one electron and acceptors that take two, or vice versa.
More chemically flexible.
Exactly. And their reduction potential, Ied agrees, isn't fixed like an annotation. It varies quite a bit, depending on the specific protein environment within the flavoprotein. This allows for fine-tuning of electron flow in different pathways.
Okay, thousands of reactions, ATP providing energy, electrons flowing through carriers. How does the cell keep this incredibly complex system running smoothly? How is it all regulated?
That's the million-dollar question. It's not just isolated pathways. It's a highly interconnected network, a meshwork. Think about glucose 6-phosphate again.
Right, it can go into glycolysis for ATP or the pentose phosphate pathway for NADPH or be used to make glycogen.
Exactly. The cell has to constantly make decisions about resource allocation, and these decisions ripple through the entire network. It's about maintaining balance.
Not equilibrium, you said, but a dynamic, steady state.
Precisely. Homeostasis. Keeping internal conditions relatively stable even when the outside world is changing. Substrates enter a pathway, intermediates are processed, products leave, all at matching rates, so concentrations stay fairly constant, though they can shift based on needs.
And if that balance fails,
That's often the basis of disease. Diabetes, for instance, is a failure of glucose homeostasis.
so how does the cell achieve this balance? What are the control mechanisms?
Regulation happens at multiple levels and on different timescales. Broadly, you can control either how much enzyme you have or how active the existing enzyme molecules are.
Controlling the amount that sounds like genetics, controlling protein synthesis.
Yes. Adjusting gene expression is a major way. Hormones or other signals can trigger transcription factors to turn genes for specific metabolic enzymes on or off. This is relatively slow, hours to days. You can also control how stable the messenger RNA is, how fast it's translated, or how quickly the enzyme protein itself gets degraded. Cells have sophisticated protein breakdown machinery, like the ubiquitin proteasome system.
And putting enzymes in different parts of the cell, compartmentalization.
That's another key strategy. Separating opposing pathways, like fatty acid synthesis in the cytosol and breakdown in the mitochondria, prevents futile cycles.
Okay, that's controlling enzyme levels. What about controlling the activity of the enzymes already there? That sounds faster.
Much faster, milliseconds to seconds. The simplest factor is just substrate availability. If the substrate concentration is low, the enzyme works slowly. More sophisticated is allosteric control. Small molecules, effectors, bind to the enzyme at a site different from the active site.
And this changes the enzyme's shape and activity.
Exactly. It can activate or inhibit the enzyme. Often these effectors are downstream products of the pathway, providing feedback inhibition, or indicators of the cell's energy state. Allosteric enzymes often show sigmoid kinetics, meaning small changes in effector concentration can cause big shifts in activity, like a sensitive switch.
What else?
Covalent modification is huge. The most common type is phosphorylation and dephosphorylation. Adding or removing a phosphate group, catalyzed by protein kinases and phosphatases, can dramatically alter an enzyme's conformation and activity. Hormones often work this way. And finally, some enzymes are regulated by binding to specific regulatory proteins.
It really is like an orchestra. So which steps in a pathway are usually the ones being controlled? Is it random?
Not at all. The key control points are typically enzymes catalyzing reactions that are far from equilibrium inside the cell. Reactions with a large negative actual AA.
Why those ones?
Because reactions near equilibrium, close to zero, are easily reversible by small changes in substrate or product levels. They respond quickly, but don't offer strong directional control. But a reaction that's strongly exergonic, far from equilibrium, acts like a valve. Controlling the enzyme that catalyzes this step provides effective control over the flow through the entire pathway. Think of phosphofructokinase 1, PFK1, in glycolysis, a major control point.
And ATP keeps coming up. It must play a role in regulation, too.
A massive role. As we said, cells keep ATP levels high, far from the equilibrium of its hydrolysis. The ratio of ATP to ADP and AMP is a critical indicator of the cell's energy status, its energy charge.
High ATP means high energy.
Right. And interestingly, the concentration of AMP is an even more sensitive indicator of energy stress than the ATP-ADP ratio.
Oh, so?
Because of that adenylate kinase reaction, 2-ADP, ADP plus AMP. If ATP levels drop just a little, say 10%, ADP levels rise. And because of the equilibrium, AMP levels can shoot up dramatically, maybe 500% or 600%.
Wow, a tiny dip in ATP causes a huge spike in AMP.
Exactly. Which makes AMP a fantastic signal for low energy.
Yeah.
And this brings us to a master regulator. AMP-activated protein kinase, or AMPK.
The cell's energy sensor.
That's the perfect description. When AMP levels rise, AMPK gets activated. It then phosphorylates numerous downstream targets. Essentially, AMPK switches on pathways that generate ATP, like glucose uptake and glycolysis, fatty acid oxidation, and switches off pathways that consume ATP, like synthesizing glycogen, fatty acids, cholesterol, proteins.
It acts like a central controller trying to restore energy balance.
Precisely. It's a crucial survival mechanism found across eukaryotes, maintaining metabolic homeostasis.
We have definitely covered a lot of ground today, from the basic laws of thermodynamics applied to life.
Through the specific chemical tools cells use, like group transfers and redox reactions.
Focusing on ATP as that central energy currency.
And the elegant ways electron flow is managed by carriers like NAD and FAD.
All the way to the incredibly sophisticated web of metabolic regulation, keeping everything in that dynamic, steady state.
It really highlights the elegance and efficiency shaped by evolution. How thermodynamics is harnessed, how specific molecular mechanisms like phosphorylation or allostery orchestrate everything. It's quite amazing when you unpack it.
This deep dive into Lehninger Chapter 13 really brings it home. Life is fundamentally a chemical system, constantly managing energy, maintaining order, adapting. It clarifies so many of those complex pathways and structures.
So the next time you eat a meal, go for a run, or even just sit thinking. Maybe pause and consider the sheer scale of coordinated chemistry happening inside you. Billions upon billions of ATP molecules being made and used, electrons flowing, enzymes being switched on and off.
It makes you wonder, what other complex biological phenomena, maybe even things like aging or certain diseases, could be better understood by digging deep into these fundamental biochemical principles? Food for thought. Thank you for joining us and being part of the Last Minute Lecture family. We hope this deep dive helps you see the chemistry of life in a new light.